Welcome everyone to today's MPCCC Precision Cancer Seminar. I'd like to acknowledge the traditional owners of the land in which we join together today and pay my acknowledgements to their elders past, present and emerging, as well as to any members of the Aboriginal or Torres Strait Islander community gathered with us this afternoon. As you would all probably know, the MPCCC Precision Cancer Program is a community of scientific researchers, health professionals and academics from across Victoria with an interest in precision medicine. The program is working to establish better access to molecular sequencing for cancer patients across southeast Victoria. And we also host several tumour-specific molecular tumour boards to support the interpretation and clinical application of molecular data. The MPCCC Precision Cancer Seminars are an important part of this program. They help to share the latest research activities across the partnership in Victoria and internationally, and to communicate new ideas and approaches in clinical care. So today, as you can see, I'm really pleased to welcome Nick Huntington as our guest speaker. Nick may choose to elaborate on what I'm about to say, but my notes in introduction have some of the key aspects to Nick's background, and that is an NHMRC Leadership Fellow and Head of the Cancer Immunotherapy Laboratory at Monash Biomedicine Discovery Institute. He's an international opinion leader on natural killer cell biology and immunotherapy and has won several awards for his work in this field. He currently leads the research program aimed at deciphering and the regulatory network that controls NK cell tumour immunity. He's the former president of the Melbourne Immunotherapy Network. He's a co-founder and chief scientist of Innate Proprietary Limited. I hope that's how you pronounce it, Nick. So without any further ado, I'll hand over to you, Nick. You can correct anything I've said or add to it if you'd like, but we're all very much looking forward to your presentation. Thank you.
Thank you. No, that was all as accurate as it can be. Let me just grab a pointer, laser pointer. There we go. You can see the laser pointer, all good?
Can, thank you.
Right, I'm gonna, actually, I'm just gonna get rid of my my screen where that's it. All right. So thanks for the introduction and thanks for the opportunity to share some of our lab's work. What I'll talk about mostly is probably last 10 years of work in really trying to understand the role of natural killer cells in contributing to immunity to cancer, or the endogenous immune response to cancer. So with my disclosures, as you said, the founder and CSO at Onco, and we have collaborations with Gilead and Kite, and I sit on a BMS scientific advisory board as well. So the cancer immunotherapy revolution has really given us great insight into targeting largely the adaptive immune response for really pronounced and prolonged, I guess, immunity and control of solid tumors. And this is just an example here of late stage melanoma, where we've seen, from chemotherapy to targeted therapies, slightly increasing response rates. The big sort of shift in treatment was the advent of immune checkpoint blockade therapies, where now we're targeting endogenous immune cells and T cells to really have a durable control of tumor. And that's given us, up to sort of, 40-odd% response rates in these late stages for 5-year survival rates. And I guess the next big question is, or the quest is, how do we increase that with combinations of, immune checkpoint blockade along with genomic or chemotherapy or radiation therapy. So what's become very clear is you can target, T cells with combination immunotherapy towards a number of checkpoints on the surface of T cells to allow them to continue to respond to tumor antigens. You can also engineer T cells to recognize tumor antigens using chimeric antigen receptor technology. And you can also inject in bispecifics or biologics, which act as a bridge between the tumor and the T cell to also induce a sort of artificial T cell control of the tumor in a very antigen specific manner. The PD-1 axis and PD-L1 axis is the major pathway that's been sort of investigated in cancer immunotherapy, and this is just a graph from the CRI to highlight that combination trials looking at enhancing T cell responses through targeting PD-1 and PD-L1, coupled with other agents such as targeted therapies, chemo, radiation. And you can see up here that the number of trials that have been started, per year is significantly increasing with sort of chemo and combination with VEGFR over time. And there's 6,000 sort of trials ongoing from late last year. So there's a lot of bets really placed on PD-1 and T cells in having durable responses and improved response rates in cancer. So as I said, the current cancer immunotherapy approaches are largely T cell centric. Most combos, as I said, focusing on T cell checkpoints, which really is the same axis. It's enhancing T cell recognition of the tumor and fitness and continual lysis of tumor cells by T cells. We focus on the innate immune system and largely natural killer cells. So the innate immune system can spontaneously respond to malignancy. It really is a first line of defense. There's myeloid cells that might recognize pattern-associated molecular patterns, and PAMPs, which are similar sort of danger signals that might be happening when a tumor cell is undergoing transformation or cell death. Myeloid cells will drive a lot of inflammation into tumors, but also phagocytosis of tumor cells. Natural killer cells, which, as I said, we focus on, can also recognize tumor cells in a non-MHC restricted manner, and they directly kill cells. Tumor cells would also drive a lot of inflammation, and I'll talk about this in great detail. There's also dendritic cells, which are essential for the presentation of antigen and in the draining lymph node to T cells, and also drive inflammation. So the innate system acts as that sort of first step, detection of tumorigenesis, initiation of inflammation, and then sort of potentiation of a T cell response, which in generally is controlling the tumor. So it really acts as a bridge to the adaptive immune response. Natural killer cells specifically, they're really sort of an innate equivalent to CD8 T cells, which are the focus of all of the immune checkpoint blockade therapies. The NK cell development and function is dependent on the cytokine IL-15, which I'll spend a lot of time talking about later in the presentation. NK cells are activated upon detection of altered self. So this is, infection or transformation, DNA damage response in cells. Natural killer cells can detect that. They also can detect changes in class 1, so downregulations of MHC class 1, and they do this by an array of germline encoded receptors. I guess this absence of MHC is called missing self, and that's been exploited sort of heavily in hematological malignancies to induce graft versus leukemia responses in hematopoietic stem cell transplants. But in general, effective NK cell therapies are yet to be realized. So, do NK cells infiltrate solid tumors? Yes, they do. I think when you look at flow cytometry of a metastatic melanoma, you see that NK cells, are a minor component of the infiltrate compared to T cells. But 100% of those NK cells do have activating receptors to melanoma ligands, for example, receptors like NKp46 and NKG2D, which recognize stress ligands on melanoma cells. Whereas, very few of those CD8 T cells are actually neoantigen specific. So I'd say, pound for pound, there's more antigen specific NK cells or melanoma specific NK cells than antigen specific T cells. And one of those receptors, NKp46, is actually really important for melanoma metastasis immunity. So, for example, if we remove or delete the NKp46 receptor, so all of the natural killer cells are normal except they're missing just one receptor, you pretty much lose all of your immunity to melanoma metastasis. So there are ligands on this melanoma which are recognized by NKp46 and allows NK cells to eradicate those metastases. This is just a slide of a colorectal cancer model, MC38, where in GFP we're staining with staining for NK cells with a NCR1 GFP and in purple CD8. And I just highlight this to say that there's been a bunch of studies that actually show that when you're looking at histology, you get a very different picture to what you get when you look at sort of flow cytometry, and even looking at histology and flow from the same tumors, you can underestimate, certain immune cells contribution by up to sort of 10-fold. So when I say NK cells are a minor component, by flow they seem to be a minor component, but when you start looking at histology, they seem to be much more prevalent than we suggest from flow cytometry. So is there evidence that NK cells now impact on tumor immunity? This is a question that we had early on, and to make sure all of our studies are actually relevant. So we looked at the impact of NK cell infiltration on the outcome of solid cancers because it had been really poorly documented. We defined using single cell RNA sequencing technologies from a bunch of different papers and different data sets, as well as our own. We defined a signature for NK cells. So these are genes that are highly expressed in NK cells versus other immune cells and not expressed in tumor cells. And then when you look at the Sloan Kettering cutaneous melanoma data set, what we see is there's patients that have a low expression of the NK signature, and there's patients that have a high expression of the NK signature. And when you plot melanoma survival using the high signature versus the low signature for NK cells, we can clearly see distinct outcomes based on NK cell infiltration. So, we see that NK cells do aid tumor immunity, at least from our analysis of the TCGA data set. And then we sort of set out to really define what are the roles of NK cells through each sort of stage of tumorigenesis. And we came up with this sort of idea of the cancer natural killer cell immunity cycle, which we published a couple of years ago. But basically, I'll take you through each step in this talk, from NK cells actually recognizing the stress ligands, to how tumors might escape, to how NK cells control metastases, how they drive tumor inflammation, how they potentiate T cell responses, how they aid immune checkpoint blockade responses, and then potentially how you can target NK cells when in immune checkpoint blockade resistant or immune checkpoint blockade non-responding tumors, or even cold tumors. So we'll try to get through most of this with examples of where NK cells play roles in the cancer immunity cycle along the way. So the first one really is the NK cell recognition. So that is recognition of stress ligands or early cellular transformations. You can see this is a cartoon taken from that review, but tumor cells have a number of tumor ligands that can be recognized by a number of activating NK cell receptors. They also have a number of inhibitory ligands for inhibitory NK cell receptors. They have a number of activating cytokine receptors or stimulatory cytokine receptors, as well as the array of receptors for suppressive factors. Now, what determines whether an NK cell is going to, lyse a cell or kill a cell, promote inflammation in response to a tumor, is really just going to be the weight of inputs from these tumor ligands, whether they be stimulatory versus inhibitory, and also from inflammatory cytokines. So it really is a net input of these kind of four factors, which really sort of dictate whether a natural killer cell will participate in sort of tumor lysis and release of cytokines and chemokines. That's really the key role of NK cells, is that initial detection and response to transformation. So many of these ligands are also, stress-induced ligands. Some of them are germline ligands that might be upregulated or downregulated in response to transformation, and that would detect, sort of elicit an NK cell response. And the real evidence that these receptors are important is a nice example with melanoma, as I mentioned earlier. If you delete one receptor, NKp46, you have no more control of B16, you have no more control of LWT1, even though when you look at the sort of immature, mature 1, mature 2, the types of NK cells that are present in the mouse, there's no difference in the maturation stage of these of NK cells, or the numbers of NK cells in these mice. They're just lacking one receptor on the NK cells. And you can see when you transplant these LWT1 subcutaneously that you have an impaired control and contribution to solid tumor growth as well, giving a really highlighting how important NKs are for detecting these ligands on melanoma cells and contributing to tumor immunity. The next step after they've recognized the tumor is sort of a tumor escape or a tumor immunoediting step. And here I'll represent what happens in cartoon form. You know, there's a heterogeneous pool of tumor cells. The green ones might be more sensitive to NK cells because they have higher levels of activating ligands and factors versus inhibitory ligands and factors. And then NK cells essentially shape or edit that tumor, and you preferentially lose cells that might be more immunogenic, and you end up with a tumor that has been immunoedited. Continuing on from that, these edited tumor cells can also escape, and that can be done by escape NK cell recognition and control, and that can be done through production of suppressive factors, but also cleavage of ligands that they express on their surface, such as the MIC-A and MIC-B ligands for NKG2D. And when they do that, they will evade, NK cell recognition through a number of those receptors, and you end up with this a pool of escaped or NK cell escaped tumor cells. So that's really that immunoediting and escape step that would happen next. The next step is this primary tumor outgrowth and development of a suppressive, I guess, tumor microenvironment. We spent a lot of time looking at factors that suppress NK cells in vitro and how that contributed to in vivo control of tumors. And I'll present data on two factors today, TGF-beta and activin A. So these are soluble mediators that are expressed in the tumor microenvironment and also produced by immune cells themselves. As you can see, as you treat NK cells with TGF and activin, you do upregulate SMAD2 and 3 phosphorylation. So you activate these transcription factors downstream of TGF and activin receptors. And these transcription factors then, have a number of effects on NK activity, which includes the suppression of granzyme, a key serine protease, which is important for the lysis of tumor cells. TGF will also induce a lot of NK cell apoptosis, and now we're looking at the number of live cells in the presence of TGF versus IL-15 alone, and you can appreciate a large drop in viability of the cells, a downregulation of pro-survival genes such as MCR1, and prevention of proliferation or a block in proliferation. So TGF and activin are suppressive factors that signal through SMAD2 and 3 to have a large suppressive effect, global effect on NK cell fitness and function. So we want to have a look at how this TGF signaling impacted tumor immunity. And to do this, we generated mice that had NK cells that specifically lacked the ability to receive TGF-beta signaling using a floxed allele for the TGF-beta receptor 2 over here in red. And also NK cells that had constitutively active TGF-beta using a constitutive active TGF-beta receptor conditional allele. So the NK cells are only having hyper TGF signaling in this green, or no TGF signaling in the red. And then we looked at the NK cell phenotype, and what you see in the spleen is normally, sorry, this is the liver, I think. Normally you will see conventional NK cells and some sort of tissue resident cells. And then if you have consistent high constitutive active TGF signaling, what you end up is mostly with these tissue resident NK cells. In contrast, if you don't have any TGF signaling, you don't end up with any tissue resident NK cells. You end up with mostly these conventional NK cells. So TGF drives our tissue residency program, including the upregulation of TRAIL, CD49A, DNAM, CXCR6, CD200, and decreases the transcription factor EMIS, which is really important for some of the more Th1 anti-tumor responses, pro-Th1 responses. So what happens in vivo now if you blocked TGF-beta signaling in NK cells? Well, we can see that if we blocked TGF-beta, we can see NK cells that are getting into a sarcoma. So we've injected these mice, wild type or TGF-beta receptor 2 floxed mice, which have no TGF signaling in NK cells. We've transplanted them with an MCA tumor. If you look in the tumor, the wild type mice, over half of the cells have started to differentiate towards tissue resident, whereas most of the NK cells remain conventional in the absence of TGF-beta signaling. And if you look at what effect that has on control of tumors, it's quite clear here in a small cell lung cancer model that that NK cells that can't become resident, that don't see TGF-beta, have a very good control and anti-metastatic response to this small cell lung cancer. Whereas obviously if you're NK cell deficient, you have a huge accumulation of small cell lung cancer in the liver after IV transfer. And a similar mechanism of action works on CD8 T cells. If you have strong TGF-beta receptor 2 signaling on T cells, then you have suppression and sort of outgrowth of tumors. But if you prevent T cells from responding to TGF-beta, you can have perfect control, almost no tumorigenesis, no tumor formation in a subcutaneous model. And if you delete CD8 T cells in green, then you restore the ability for those tumors to grow. So you have a very clear effect of TGF-beta signaling suppressing NK cells and T cells. And I talk about this residency because it is actually seems to be quite important in humans as well. So we looked back at our single cell gene signatures across different cancer sets, and we looked at what we just defined as a tissue resident or a sort of an exhausted, more of an activated or a conventional NK cell signature. So we generated data sets on these resident versus exhausted signatures based on CD8 cells, CD4 cells, and also NK cells in different data sets, colorectal data sets, and also normal healthy lung. So the resident signature being in orange here, and the sort of exhausted signature being in blue. And when you start splitting infiltrated tumors into those that have a high residency and a low exhausted across different data sets, you see that the high residency is actually sort of detrimental. Those patients have a poorer outcome compared to those that have a sort of low residency and high exhausted. So again, these TGF-beta programs which drive tissue residency seem to also be relevant in humans as well. You see that exhaustion is detrimental to tumor control. The next part is, as these tumors are progressing and suppressing NK cell response, there's always metastatic tumor cells extravasating and seeding other organs. And we've studied this deeply, and we generated, probably was that 8 years ago now, maybe almost 8 years ago, we generated a black 6 mouse that specifically lacked NK cells by deleting a survival gene MCL1 using a specific CRE that's only expressed in NK cells. So we made B6 and BELB-C models where they only lacked NK cells, and then we looked at some metastatic tumor models, and here it was really very striking that if you injected in very few 50,000 B16 cells, you don't have many metastases in a normal mouse, but these mice actually started to present with even breathing difficulty at day 14, where they had a huge burden of metastases in the lung. And similar in the small cell lung cancer models, if you have a mouse that lacks CD8 T cells, there's no effect on metastases. But if they lack NK cells and you inject these tumor cells intravenously, NK cells are really efficient at detecting and eradicating these cells. And if you have no NK cells, then you do have a large burden of metastasis in the liver. Those are experimental models. We were also interested in looking at something a bit more physiological. So we looked at EO771, which is a triple negative breast cancer, when we implant that into the mammary fat pad. And what we found is in this setting, under these conditions, this work was done at WEHI, we didn't see any difference in the growth of the primary tumor over time between a wild type or an NK cell deficient mouse. But if we surgically resected these mammary tumors and waited a couple of weeks and then took the lungs, we could see that the wild type mice had largely prevented metastases to the lungs, whereas the NK cells had again a significant burden of triple negative breast cancer metastasis in the lung. So again, as the primary tumors are growing, metastatic cells are extravasating and getting into the lung. NK cells can detect and eradicate these as they extravasate in circulation, as they seed the lung. We're not exactly sure, but if you don't have NK cells, then you are going to end up with a significant burden of metastatic cells. And NK cells really have a unique and role in anti-metastatic immunity. A last model I'll share, because I think it's a really, again, sort of more physiological model, a really cool model, was developed by a colleague Thomas Gebhardt at Melbourne University, but that's where they have, again, a melanoma cell line that's put in Matrigel and just sort of just added to the epidermis of the skin. And it sort of represents, you know, the stages of human disease. You can have sort of progressive disease where you end up with metastases in the draining lymph node. You can have stable disease where the tumors will stay and be controlled by the immune system, and you can have mice that reject the tumor. So it sort of looks, you know, similar to what we having to humans when they have sort of cutaneous melanoma. But if you start looking at the draining lymph node after surgically removing these tumors, very few metastases in normal mice, and again, much significant increase in sort of tumor outgrowth or controlled metastases in the draining lymph node if you've depleted NK cells with an antibody, or if you've genetically depleted NK cells using the MCL1 floxed NK Cree. So again, there's a lot of different models there just to show that NK cells are really unique in their way that they can detect and eradicate metastatic cells, and it really is the unique property of NK cells. The next part is really how NK cells drive tumor immunity. They drive, I guess, tumor inflammation. And, you know, we, again, spent a bit of time, you know, looking at different solid tumor models and how NK cell presence or absence contributed to tumor control. And I think early on, I must admit, early on, most of the models, I guess at WeHi, didn't really show huge differences. There were a couple of models where NK cell deficient mice, the tumors grew faster and larger. And it seems to be model dependent. Now that we've moved to Monash, I think our animal houses are here a bit dirtier, which means that the immune system's a bit more active, and the NK cells are a bit more active, and therefore the lack of NK cells actually results. We see sort of clearer differences between NK deficient mice versus control mice. So this is a very, you know, popular model, MC38, colorectal model, anti-PD-1 responsive model that most labs use. And you can see in the NK deficient mice, the tumors grow, you know, more rapidly, and the NK deficient mice have a lack of the tumors have a lack of dendritic cells, and also T cells, CD8s and CD4s. You see a strong, you know, correlation between the presence of NKs and dendritic cells. If we start to look at dendritic cells versus NK cells, we see a very clear correlation between those cell types. And several studies have shown that dendritic cell recruitment to the tumors is mediated by NK cells through the production of XCL1 and 2, and then also NK can provide factors such as FLT3 and GM-CSF to enhance the maturation and function of dendritic cells. So likewise, if you look at now at a dendritic cell, CDC1 deficient mouse, again, if you've got no dendritic cells to present antigen to T cells, then you've got very little control of these MC38 tumors, and again, very little NK cell and T cell infiltration. Next, we'll look at how NK cells and the presence sort of primes and promotes T cell immunity to these tumors. What we found is that in several of our NK cell deficient models, we see impaired TNF positive CD8s in those tumors, also interferon gamma positive CD8 T cells within the tumors. And here we can see in histology as well that in brown are CD8 T cells. And again, we see a large sort of desert of CD8s within NK cell deficient tumors. And there is, you know, reasonable data showing that NK cells are a key producer of RANTES, CCL5, which is important for recruitment of NK cells, but also CD8 T cells to the tumor. So again, NK cells really promoting the polarization and function of CD8 T cells in the tumor microenvironment. So next, if NK cells contribute to in the T cell infiltration, then, you know, we asked the question of how does their presence or absence dictate response to immune checkpoint blockade. And not surprisingly, we found that NK cells are required for optimal anti-PD-1 responses. So again, we used NK deficient or sufficient mice where we had injected in MC38 tumors and treated with anti-PD-1. And you can see in the red at the bottom here is the wild type mice treated with PD-1, where you see a regression control of the tumors compared to non-treated, and NK cells treated with PD-1 or just treated with PBS in this case had no effect or no control or no effect of PD-1 on tumor control in the absence of NK cells. And again, you can see that PD-1 treatment induce, you see, an increased sort of NK, CD8 and CD4 dendritic cell, whereas the NK deficients had very poor, again, infiltration of dendritic cells, and PD-1 treatment really did nothing to promote dendritic cell or CD8 T cell responses. So again, very clear that NK cells are required for optimal responses to PD-1, at least in these MC38 mouse models. Mouse responses to immune checkpoint blockade is heterogeneous, much like humans. And this, if you inject the same tumor collaterally on either side of the mouse, those tumors typically behave the same. So if the left side doesn't respond to PD-1, then the right side doesn't. If the left side does, the right side does. So this was a nice study by Rachel Zemek in Western Australia. So it actually allows an opportunity to understand the mechanism of action of some of these therapies. And what she showed was very similar to what our findings are, is that if you put tumors on either side of the mouse, now this was a AB1 or a Renka tumor, so a mesothelioma or a kidney cancer. If you put these tumors and allow them to grow to a certain size, you can excise one of those tumors and analyze it, and then you start treating with, with anti-CTLA-4 and PD-1, so immune checkpoint blockade. And then you can see what happens to the mice. Do they respond or do they not respond? And what do we know about what the tumor infiltration, tumor microenvironment looked like in the ones that respond versus the ones that don't respond? And her conclusion was quite clear that in both models, the responders had a higher infiltration of NK cells at the time of immune checkpoint blockade treatment, and a higher proportion of activated NK cells. So NK cell frequency and activation and fitness was a dictating factor in response to immune checkpoint blockade in these mouse models. And the group of Kevin Barry and Max Krummel showed that was some similar finding in a human data set. This is a small cohort of melanoma patients being treated with PD-1. And again, the key factor that distinguished responders and non-responders was the responders had an increase in NK cells and dendritic cells. There was no real difference in Tregs or CD8 T cells in responders and non-responders. It was really the NK cell frequency that in the tumors which correlated to anti-PD-1 responses. So it establishes a sort of model where NK cells are recruited into the tumor microenvironment, and when they see tumor ligands or upregulated tumor ligands, and also growth factors such as IL-15, IL-12, and 18, they become activated. They can degranulate and generate tumor ligands. Those tumor ligands can be taken up by dendritic cells. NK cells secrete chemokines that recruit in dendritic cells as well. These dendritic cells can then go to the tumor draining lymph node, present to CD8 T cells, who can then proliferate and be recruited back to the tumor to undergo T cell mediated killing of that antigen specific in an antigen specific manner. So NK cells, drive the tumor inflammation and potentiate CD8 T cell immunity and immune checkpoint blockade response. So with what have we got, about what, 10 minutes or so? With the last point, I'll just say that, so we've gone through and seen how NK cells are important for, tumor infiltration and immune checkpoint blockade responses, but there are tumors that, don't get infiltrated, cold tumors, or tumors that don't respond to immune checkpoint blockade. And one of our, questions in the lab is, how can we target these tumors to cold tumors or non-responsive tumors to become sensitive to NK cell intervention and eventual response to immune checkpoint blockade. So I guess the field is trying to address this in a number of ways. There's, tumor antigen targeting biologics, antibody drug conjugates, antibody cytokine biologics, cell therapies, I guess, to try to ignite inflammation in cold tumors. There's also work around combinations of radio, chemo, and targeted therapies to promote tumor inflammation. But what we've focused on is a novel sort of targeting of the DNA damage response in cold tumors to try and improve NK cell recognition. Now, the model that we're going to talk about in detail is melanoma models. There's a human and mouse melanoma model we use a lot to look at this, and these models, these tumor lines, express high levels of MDM2 and 4, and they're expressed highly in most human melanomas, and it leads to a reduction in p53 expression. Now, through some in silico screens that we won't have time to go into today, we discovered that sort of combination of targeting WEE1 and AKT led to a reduction in MDM2 and a re-expression of p53. So with that data, we've used I guess there's AstraZeneca's compounds. The one I'm just going to call it MK, which is really the WEE1 inhibitor. That it's a selective potent ATP competitor for WEE1, and AstraZeneca's 5363 or AZD, which is an inhibitor of AKT123. And when you treat melanoma cells, you can see untreated have very little p53, but as you treat with doxorubicin or combinations of WEE1 AKT, you can start to increase p53 expression. We've made some p53 knockout clones of these melanoma cells to show that to use later on to validate some of the essentiality for p53 expression. We see that the B16s as well, when we start to treat with the AKT inhibitor, but also in the combination of the WEE1, we see a lot of B16 death in vitro. And again, if those cells don't express p53, we don't get any death of those B16s. Again, this 1205 Lu human melanoma cell in a xenograft grows, grows quite well. But if you treat those mice with the combination of the WEE1 AKT inhibitor, you get control of the tumor, and you only get that when the tumor expresses p53. The p53 knockout tumors cannot be controlled by this WEE1 AKT combination. And part of the mechanism of control is it does induce a lot of cell death, and especially immunogenic cell death, which sort of activates the immune system, and it can be detected by an increase in calreticulin in response to the combination here on B16s, but not in the absence of p53. So targeting WEE1 and AKT induces p53 dependent immunogenic cell death. And you can see across a number of human tumor lines, when you start to induce this immunogenic cell death, you start to upregulate ligands for NKG2D, which is an activating receptor on NK cells. So this is ULBP2, 3, MICB, ULBP1. They all increase in the presence of the inhibitor for WEE1 and AKT, but you don't see increases when those same cell lines are deficient for p53. And if you treat these tumor cells, these Lu, 1205 Lu, with a combination of the WEE1 AKT inhibitor and then co-culture them with NK cells, you can see a clear increase in NK cell killing with the cells that have been treated with the inhibitors, likely due to their increased immunogenicity via upregulation of these p53 dependent sort of stress ligands to NKG2D receptors. So most importantly now is if you go into B16 model, in vivo, this model is quite resistant to PD-1. It's poorly infiltrated due to low levels of chemokines such as CXCL9 and 10. And B16 tumors do not respond to PD-1 therapy. So we tried to say whether we could drive any kind of tumor inflammation using the AKT and WEE1 inhibitor and see whether that was synergized with PD-1. And what you can see here, and I think maybe I've cut the legend out, but untreated in gray, p53 untreated in pink, and then the WEE1 AKT inhibitor alone in purple, and the combination of the WEE1 AKT and anti-PD-1 in red. And you can see that you've prolonged the survival in the presence of the PD-1 and the WEE1 AKT. So you've been able to make these B16 sort of cold tumors. You've been able to induce a response to anti-PD-1, and that's characterized by a large increase in NK cell and activated NK cell infiltration, and also dendritic cell infiltration. And things like CXCL10, RANTES, CCL2, IL-2. So some of the, PD-1 here, so some of the factors that are required for infiltration and some of the markers of activated T cells. So you've really driven a potent sort of T cell response by driving up NK cell activity and dendritic cell recruitment in these immune checkpoint blockade response or non-responsive tumors. We went on to show that these models are, again, dependent on p53. So if you remove p53, you don't get any triple therapy response. If you remove NK cells, you don't get triple therapy response. And if you remove CD8 T cells, again, you don't get any therapy response, which is totally expected. And again, combinations of PD-1 plus the WEE1 AKT inhibitor drives a lot of NK cell infiltration and activated CD8 T cells. So what do we got? 1 minute, I think 1 minute left. So I think I'll sum up, and I won't present any of the extra stuff. So I'll sum up just by saying, like, as I've mentioned, NK cells have a clear contribution throughout many distinct stages of this cancer NK cell immunity cycle. There's contributions at most stages, and now we're trying to look at ways of increasing NK cell infiltration into cold tumors to try to potentiate immune checkpoint blockade responses. And I think we've demonstrated that we can do that with inhibitors to the WEE1 AKT pathway. There's also inhibitors to the MDM2 and 4, which is highly expressed in melanomas. There are inhibitors to that, to those proteins, but I think unfortunately it's quite toxic. There hasn't really been able to manage the toxicity of those inhibitors, and that's why it really promoted us to look at the combination of WEE1 and AKT to try to reduce MDM2 and 4 levels and promote p53 mediated immunogenic cell death of the melanomas and subsequent immune control. So with that, I think I've highlighted that there are key roles for NK cells at most stages. I won't go into this per se, but just to say that we're trying to target NK cells in vivo, and a lot of our work focuses around making NK cells more sensitive to IL-15 and IL-2, and IL-15 and IL-2 therapies have really been investigated in cancer for a long time, since the early 80s, where some patients showed durable response following treatment of these IL-2 at least, and was quite toxic, and it really failed to take off just because of the toxicity and the low response rates. But those patients that did respond have responded durably, up to 30 years later, and that sort of made IL-2 therapy get approved in the early 90s for renal cell carcinoma, in the late 90s for melanoma. But I think the toxicity for that has really sort of motivated us to try to discover novel pathways to get activate NK cells, make them more sensitive to cytokine without actually adding in exogenous cytokine. And why I don't really have time to go in it today, we've identified a negative regulator of IL-15 signaling directly in NK cells. And now we're in the process of drugging that pathway with small molecules to try to increase the responsiveness of NK cells and T cells really in the tumor microenvironment, not systemically like what would happen if you injected in IL-2 and 15, but really within the tumor microenvironment, because that's where you have high amounts of IL-15 due to inflammation, or IL-2 due to T cell activity. And if we can localize the response or the hyper responsiveness of cells to the tumor, we're likely to get a better signal without having the sort of dose limiting toxicity that we've seen with some of these systemic therapies, IL-2 and 15. So I'm going to skip through all this, and I'll just get to the points to really take away from today's talk, and hopefully got some good time for discussion. I'm keen to discuss with all of you around potential areas to collaborate. I hope I've given a flavor for the types of models that the lab works on and the areas that we're interested in. And we're very much open to collaborations with people in Melbourne. And then summarizing, so NK cells play a distinct role throughout the cancer immunity cycle. The NK score, and particularly, a greater exhausted versus residency score, predicts beneficial cancer patient outcomes. NK cells drive tumor inflammation through factors such as RANTES and XCL1 and 2 to potentiate CD8 T cell responses. NK cells uniquely detect, metastases. I've written here melanoma metastases, but it's most metastatic models that have been investigated are their detection and eradication is dependent on NK cells. Restoring p53 in melanoma drives NK cell infiltration and restores immune checkpoint blockade in these cold, non-responsive tumors. I didn't go into it, but there are intracellular checkpoints in tumor-resident NK cells and T cells, which we're drugging, and obviously TGF-beta being a very suppressive factor for NK cell function. And we do believe that efficient targeting of endogenous NK cells can improve tumor outcomes. So with that, I'll just thank a lot of people that did the work over the last sort of 10 years, and Rebecca, Del Conte, Fernando Guimaras, Momene, Hei Yong, Jay and Joe over at Onco. The CIS stuff's a collaboration with Sandra. Gavin Robinson was a collaborator for the WEE1 AKT work, Thomas Gebhardt on the melanoma models. I've had good interactions with Mark Shackleton and David for human samples and sort of discussion of clinical relevance of NK cell targeting in human tumors. And with that, I will stop the share and take some questions.
Great. Thanks so much, Nick. Amazing. Now, it's only there's a smallish group online. If you have a question, I suggest you just raise your hand and turn your mic on. And Nick, you were quick to.
Otherwise, I'll keep presenting.
Yeah, we can flip back to that. Nick, do you want to turn your mic on and actually ask?
My mic's on. Can you hear me?
Sorry, Nick Wong. I'm sorry.
Oh,
sorry, I was just clapping. Not putting my hand up.
I can't even. Oh, yep. Nick's frozen. Or is that just a photo?
There he is.
Hey, Nick, how are you? Nice to see you.
Yeah, likewise. No, brilliant talk.
Thank
you. My mum said the same.
Nick, I wondered if you could just orientate us back to one of your first slides where you talked about the diversity of clinical trials that are being undertaken at the moment, and perhaps having heard your talk, reorientating us to where your work actually pictures along that landscape.
Yeah. So yeah, I didn't have a lot of time left. I wanted to get to the questions. I didn't really want to get into how we're targeting the cytokine checkpoints, but basically, in terms of, we're still focused much on targeting the immune cells themselves, and all of those trials are targeting, PD-1 plus something else. So trying to activate the T cells, but then trying to make the tumors potentially, undergo more apoptosis, or having growth defects, suppression of growth on the tumor cells, hitting sort of other pathways outside of T cells. A lot of the work of the trials are looking at other pathways besides T cells. We're very much focused on immune cells and combining things like PD-1 with negative regulator or blockades of negative regulators of cytokine responses. So if we can make T cells and NK cells more responsive to cytokines within the tumor microenvironment, it sort of lowers their threshold to be required, lowers their threshold for activation by a neoantigen. It sort of reprograms them metabolically as well, allows them to persist and undergo more proliferation, but also more killing and resistance to suppression. And it doesn't require a checkpoint ligand, for example, on a tumor cell. So PD-1 is going to be effective if there's PD-1 ligand, but if there's no ligand, then PD-1 blockade has no real impact on T cell control. So the cytokine checkpoints allow you to actually make the whole pool of T cells more responsive, more activated, fitter. But those that see neoantigen will have a lower threshold to be activated and have a better response. And we've seen that. I could probably share some data. Maybe I'll just share one slide then, actually, just to quickly to give you an example of that. But yeah, for example, I'll go full screen. For example, on this, for example, on this slide here. So if you treat this SM1 WT1 tumor, if you treat them with PD-1, there's no response. But if you delete this intracellular checkpoint, so this negative regulator of IL-15, and then you treat with PD-1, you do get a response. And this is the area where we think that there's an untapped sort of potential for regulating cytokine responsiveness and metabolism of infiltrating lymphocytes. And I feel that that's where you'll see, synergy with T cell blockade or T cell immune checkpoint blockade, without having to hit more inhibitory ligand receptor interactions, which most of the field are doing. So combination of LAG3 inhibitor and PD-1 inhibitor has just been approved. But again, they're sort of targeting, receptors to block sort of immunosuppression via ligands. Whereas where our approach is really trying to increase the fitness and metabolism of all T cells and NK cells within the tumor.
This slide specifically says solid tumours. That was
yeah. I think
my
that's
follow-up question. A lot of your work was you showed kidney and melanoma, but I don't mean expanding in a negative way, but applying this more universally. Where do you think that is? That just a hope, or do you think there's a logical way forward to?
Yeah, I mean, our strategy would be to target inflamed tumors where there are clear NK cell and T cell infiltration. The breadth of that is wide. MSI positive CRC and non-small cell lung cancer, triple negative breast, head and neck squamous cell carcinoma. Melanoma is probably a hard one to get into because there's already pretty effective immunotherapies, but again, most patients wouldn't respond to combination immune checkpoint blockade in melanoma as well. But it has to be a tumor that has is immunogenic and has a T and NK cell infiltration component. Obviously focusing on solid, because focusing on trials on metastatic disease are not going to happen.
Mark Shackleton was online, but I
had a chat with him yesterday actually about this this very issue. So it's okay.
I think he may have had to dash, which is one of the reasons I'm speaking and not him just at the moment. But
no problems. I think the major point of making cells more sensitive to things like IL-15 and IL-2 is it does synergize. It sort of makes them more resistant to immunosuppression as well. So we definitely see that reduced ability for TGF-beta to suppress T cells when they are deficient for cis, where they're more hyper responsive to IL-15. So our small molecule program is really around inhibiting the degradation of the IL-15 and 2 receptor to have higher levels of those receptors and maintain good good T cell and NK cell responses and fitness within tumor microenvironments. There's Renee Taylor. Thanks for the fantastic seminar. Oh, thanks, Renee. You're welcome.
Okay, well, at 4 minutes to the hour, I see people are beginning to leave. Nick, I think it's up to me to thank you very much for a very stimulating presentation. I hope some of the conversation perhaps we haven't had right now, but we'll have in coming weeks and months as people get to think about your work and try.
Yeah. Be good to catch up with everyone in person. So if there's any, I don't know if this is a forum that gets face to face, or otherwise any other Monash cancer forums face to face, it'd be great to see everyone.
Great. I think Renee's actually has left the meeting, but she is hoping, along with Sefi, I think, to have some face.
I think we have a meeting. Yeah, we have a cancer retreat organised, I think, for Huntington Golf Club later this year. That'll be good.
Excellent.
If you're all there, it'd be great to catch up.
Great. Well, thank you so much, Nick. I guess.
Yeah, thanks for your attention.
Thank you. Just to say to everyone else who is actually, I can see the numbers are dwindling. Our next seminar is anticipated to be in August. We're hoping to have one in July with a special feature, which Anna and I are still working on in the background, but we'll announce that via the normal means when we've got it organised. So I guess thank you very much for attending. Have a nice weekend when you get there, and see you next time.
Thanks.